Hello students, welcome back to the last module of this chapter where we are going to learn in detail about nucleus and how to use microscope. So let's not waste the time and start the video.
Science. In this video, I'd like to look at the nucleus. I've always heard that the nucleus is the brain of the cell. But why is it the brain? And what are the major parts of the nucleus? The nucleus is a large organelle. In fact, you can see the nucleus with a compound microscope. Here it is inside a cheek cell. I'd like to look at four main components of the nucleus. First, the nuclear membrane, nuclear pores, nucleoplasm, and the nucleolus. The nucleus is surrounded by a nuclear membrane, and this is very similar to the cell membrane, and it is also composed of two layers. It protects and houses the other parts of the nucleus. You can also find ribosomes on the outside of the nuclear membrane. Up next are the nuclear pores. These are openings found on the nuclear membrane. They allow items to move in and out of the nucleus. An example would be messenger RNA.
You are heading through a nuclear pore and into the plant cell nucleus. The nucleus is home to a plant's DNA. Like meters and meters of cell blueprints, the DNA make the plant what it is.
Nucleoplasm is a gel-like or watery material found inside the nuclear membrane. You will find the DNA suspended in the nucleoplasm. The nucleolus is almost like a nucleus inside the nucleus. It has a very high concentration of DNA. It is also the site of transcription, which is the starting point of protein synthesis. This is how the nucleus controls the cell. It has all the information on how to make the stuff like proteins for the cell. These proteins are used for many purposes inside the cell. Ribosomes are also made at the nucleolus. All eukaryotic cells have a nucleus, and bacteria and other prokaryotic cells do not have a nucleus. Hope that helps.
Compound microscope. Objective: to learn about the construction and working of a compound microscope. A compound microscope has multiple lenses which collect light from the object and then focus the light into the eye. These microscopes have the ability to magnify very minute objects to a substantial size, clearly showing the smallest details. There are three major parts to the compound microscope: a body tube, an objective lens, and an eyepiece. The body tube is comprised of two smaller telescoping tubes with lenses coaxially mounted at either end. The wider tube holds the objective lens, while the narrow tube holds the eyepiece. The knobs for both coarse and fine adjustment allow the tubes to slide one into another, varying the distance between the two lenses. The lenses are convex with different focal lengths. The lens closer to the object is the objective and has a short focal length, FO, usually below 1 centimeter. The lens nearer the eye is the eyepiece. Although the eyepiece also has a short focal length, FE, it is still greater than the focal length of the objective. A revolving nosepiece, known as a turret, is attached to the lower end of the objective. The turret can be rotated to change the power magnification as necessary. Below the objective, sits a flat platform known as the stage. On the surface of the stage, two stage clips hold the slide in place. A small aperture is present in the stage, allowing light into the body. The body tube is arranged on an arm which is connected to a strong base. This arm allows the body of the microscope to tilt. A small mirror at the base of the microscope reflects the light through the stage aperture. Above the mirror, an iris, also known as a diaphragm, is present. Iris helps in controlling the amount of light passing into the stage aperture. Working of the compound microscope. The object O is placed just outside FO, the focus of the objective lens. A real, inverted and magnified image, I1, is formed. The magnified image, I1, acts as an object for the eye lens. The eyepiece is adjusted such that the image I1 lies within the focus of the eyepiece, FE. The eyepiece functions as a magnifying glass and forms a virtual, erect, and magnified image, I2, of the object. Magnification of the compound microscope. The magnifying power of a lens is expressed in diameters. A lens magnifying an object 5 times has a magnification of 5 diameters and is written as 5x.
Well, students, I guess you might have find it interesting to know about the cells that make up our body and how we can see cells through the microscope. So let's meet in the next chapter with more interesting videos. Take care.